nucleus. In the cell, eukaryotic cell, the dynamic center is a nucleus which is known as cell brain. The nucleus is discovered by Robert Brown. Later on, the nucleus is basic stained and the network is, the tiny fibrous network is called as a chromatin by Fleming. Chromatin by Fleming scientist kind that the thin thread-like network in the nucleus which is stained with basis kind is chromatin. In interface nucleus, the nucleus is widely expanded, which contains the three parts, that is nucleoplasm, chromatin and nucleolus. The electronic microscope studies reveals that the nucleus is enclosed with the nuclear membrane. Two membranes are enclosed the nucleus. So the nuclear membrane is double membrane structure which is interrupted with the fusion of nuclear membrane. As the result, the pores are formed. These are called nucleopores. So what is the use of nucleopores? It is passage for movement of proteins or RNA between cytoplasm and nucleoplasm. The nuclear membrane is a barrier between the nucleoplasm and cytoplasm. The space between the two membranes, is called perinuclear space. This is the perinuclear space. So, generally the cells are uninucleate, but in some of the organisms, this cell may have more than one nuclei. There is a binucleate, multinucleate condition. If multinucleate condition is there is a xenocytic cell. Example, Rhizopus. Vacheria. So, Vacheria, many nucleated organisms, xenocytic organisms. So, in Tapetum, dinucleated, two nuclei is present. Some of the mature cells in both plants and animals, nucleus is absent, lack of nucleus in some of the matured cells in both plants and animals. Red blood cell, enucleated. Sieve tubes in phloem, enucleated. So in nucleus, the spherical stained structure is nucleolus. This is the nucleolus. So generally, the nucleolus is non-membrane bound structure which is continuous with the nucleoplasm. It is made of the nucleoproteins. In actively photosynthesizing cells, more than one nucleolus may be present. So it produces it is the site for ribosomal synthesis. Then the fluid-like substance in the nucleus is called nucleoplasm. In the nucleoplasm, the thin filamentous network is called chromatin. So it is not clearly visible. In nucleus. During the cell division, the chromatin which is coiled and condensed into rod-like structure, that is called chromosome. During the cell division, the chromatin material which is coiled and condensed very tightly to form a rod-like structure, that is called chromosome. So the length of the chromatin is vary. For example, in human being, red blood cells or human being cells, the length of the chromatin is approximately 2 meters, which is equally distributed among the 46 chromosomes. The length of the chromatin is vary from cell to cell organism to organism. For example, in human being, the chromatin is the thread of chromatin is approximately 2 meters, which is equally distributed among 46 chromosomes. 46 chromosomes. So, in a chromosome, the non-stained constriction, a non-stained constriction is present in the chromosome, which is a non-stained constriction is present. A notch is present, which is called centromere. This is the centromere. Centromere which bind the two chromatids together. So the two chromatids are bind at the centromere. The primary constriction on chromosome is called centromere. This is the centromere. The centromere which bind the two chromatids together. The chromosome has two chromatids, two arms on either side of the centromere. These two chromatids are bind together with the help of centromere at the centromere. The centromere which is enveloped with the proteinaceous core, proteinaceous plates, that is called kinetic core. This is the kinetic core. So, based on the position of the centromere, the chromosomes are divided into 4 types. Based on the position of the centromere, the chromosomes are divided into 4 types, classified into 4 types. That is the metacentric. So, when the centromere is exactly at the center, as a result, the two arms are equal, then it is a metacentric. Just the submetacentric, the centromere is just aside from center, away from the center. As a result, one of the arm is short, another one is long. This is the submetacentric. Then acrocentric. So completely the centromere is towards near to the one of the chromosome. So as a very short arm, a long arm. This is the acrocentric. Depending on the position of the centromere, the chromosomes are classified into 4 types: metacentric, submetacentric, acrocentric. Then telocentric. So this is the centromere present at end of, telo means ends, end of the chromosome, which has only one arm. That is a telocentric. It is important for 4 marks. So classify the chromosomes based on the centromere. Write about the types of chromosomes based on the centromere. So with diagrammatic representation, like that, you have to draw this. Then in some of the chromosomes, this non-stained secondary construction is present. Which appears like a bulb or satellite, appear like a satellite. So this is called satellite. So chromosomes are called SAT chromosomes. This is a SAT chromosome. In some of the chromosomes, a non-stained secondary construction is present, which away from the secondary construction, the chromosome appears as a globe or a spherical body, which is known as a satellite. Such chromosomes are called satellite chromosomes. So further we discuss about that, the continuation of the nucleus, the nucleosomes. It is also important for 4 marks. Nucleosome. When we view the chromatin under electronic microscope, it appears beads on the string of chromatin. Beads, పూసల దండ మాదిరిగా. Chromatin ఎట్లా కనిపిస్తుంది? పూసల దండం. Beads on the string. When we view under electronic microscope, the chromatin appears as beads on the string. So like this, పూసల దండ మాదిరిగా కనిపిస్తుంది. So these bead-like structures are called nucleosomes. nucleosomes. The nucleosomes which are the nucleosomes which contain 200 base pair double helix DNA wrapped around. So wrap around. So 200 base pair double helix DNA wrap around the atomer of histone. This is the atomer of histone. This is the 8 histone proteins which are formed into unit. That is 8 histone proteins are associated to form a unit, that atomer. So 200 base pair the double helix DNA which wrap around the histone atomer of each of 2 copies of 4 histone proteins. Each of 2 copies of 4 histone proteins. In histone atomer, the 2 copies of histone proteins of 4 types are present. That is H2A, H2B, H3, H4. The atomer which is made up with the 8 histone proteins of each of 2 copies of 4 histones. So this is atomer. The histone atomer is wrapped around with double helix DNA. The H1 histone protein is and outside the nucleosome which seal the DNA. At the entry and exit with the atomer. The DNA which is present between the 2 nucleosomes. Is called linker DNA. This is the linker DNA. This is linker DNA. Thus, the chromatin is coil and condense as nucleosomes to form a coil and condense, which is in the chromatin material which is formed into nucleosomes and arranged in the nucleus, and further it is coil and condense to form a chromosome. That is the nucleosome. What is nucleosome? When it is viewed under electronic microscope, the chromatin appears beads on the string, which is known as nucleosome. What it contains? The nucleosome contains a 200 base pair double helix DNA wrapped around atomer of histone, each of 2 copies of the 4 histones, that is H2A, H2B, H3, H4. Then H1 histone protein is and outside the nucleosome which seal the DNA at the exit and at the entry and exit to the nucleosome. Then the DNA which is present between the 2 nucleosomes is called linker DNA. Thus the chromatin is coiled as nucleosomes in the nucleus. Then further it is coiled and condensed to become a chromosomes, to form a chromosomes. Thus the chromatin material is which is included, which is arranged in the nucleus very tightly with the nucleosomes. That's all. This is the nucleus. Now, what is nucleosome? when it is viewed under electronic microscope, the chromatin appears as beads on the string. That knots are called nucleosomes. The nucleosome, which has 200 base pair double helix DNA wrap around the histone of atomer of each 2 copies of 4 types of histones, that is H2A, H2B, H3, H4. The H1 histone is present and outside the nucleosome, which seals the DNA with atomer at the entry and exit. Then the DNA present between the 2 nucleosomes is called linker DNA. Thus, the positive negatively DNA and the association, nucleosome is the association of negative DNA strands and positive histone atomers are formed into nucleosomes. Thus, with the nucleosomes, by the formation of nucleosomes, the chromatin material is embedded or arranged in the nucleus. Further, the chromatin material, which is coiled and condensed to form a chromosome. Next, further, the microbodies of cell. Microbodies of cell, which are called microbodies of cell. It is also important for 2 marks. So, the peroxisomes and glyoxysomes. The membrane-bound peroxisomes and glyoxysomes are commonly called or commonly known as microbodies of cell. Microbodies. The membrane-bound peroxisomes and glyoxysomes are termed as microbodies of cell. Microbodies of cells. So these are the membrane-bound small organelles, membrane-bound spherical small organelles, which are known as microbodies of cell. The peroxisomes and glyoxysomes are microbodies of cell. What are the functions of peroxisomes and glyoxysomes? The peroxisome, which involve a catabolism of long chain of lipids. So the peroxisome which involve catabolism of long chain of lipids, which is break down the lipids. And also it involves and also it play an important role during the photorespiration to protect the cell from toxic hydrogen peroxide. It play an important role in plant cells. So it play a major role during the photorespiration protect the cell from toxic effect from hydrogen peroxide. So during the photorespiration, it play an important role. During the photorespiration, protecting the cell from toxic effect of hydrogen peroxide. Hydrogen peroxide. From hydrogen peroxide is the lethal effect to the cells. Thus, the peroxisomes are protecting the cells during the photorespiration from toxic effect of hydrogen peroxide. Then, glycosomes are rich in fat containing germinating seeds. So, glycosomes are present in which parts? So, the glycosomes are present in rich fat containing germinating seeds. Rich fat containing germinating seeds. So, rich fat containing germinating seeds. Example, castor. So, the glycosomes which have the glyoxylate cycle enzymes. During the photorespiration, the glucose, which is generally converted into pyruvic acid, but glycolic acid, which is converted, that is a glycylate acid. So, the glycosomes have the enzymes of glycylate cycle. Then it convert into fat, but they convert into fats into starch. During the glycylate cycle, the fats are converted into starch. So these are the peroxisomes. Which are peroxisomes? The small membrane-bound peroxisomes and glycosomes in plant cells. In plant cells, the peroxisomes and glycosomes are considered as a microbodies of cells. The peroxisomes and glycosomes are microbodies of cell in plant cell. What are microbodies? The peroxisomes, the membrane-bound peroxisomes and glycosomes are microbodies in plant cells. The peroxisomes which are involve catabolism of long lipid chains, and also the peroxisomes are protect the cell from toxic effect of hydrogen peroxide during the photorespiration. The glycosomes are present in fat-rich germinating seeds, castor. The glycosomes which have the glycolytic cycle enzymes to convert the fats into starch. That is the microbodies. So with this, the 9th chapter is over. That is the cell and structure. Cell is the basic life. So the cell, 9th chapter is cell structure is over. In this, we discussed about the cell types of cells, cell organelles and their functions. Next, we are going to proceed the 10th chapter, biomolecules. 10th chapter. So, in 9th chapter, the important things are, for 4 marks, cell theory, prokaryotic cell, nucleus, types of chromosomes, mitochondria, chloroplast. Difference between the rough endoplasmic reticulum and smooth endoplasmic reticulum. These are the expected 4 mark questions. So 2 more questions. Nucleosome. What are microbodies? And about the nucleus, endoplasmic reticulum, vacuoles. There is 2 more questions. 2 more questions will be asked on those cell organelles also. Cell organelles also. Today, we shall discuss about the biomolecules. What is biomolecules? Bio means life. The organic compounds which are found in the living organisms. The organic compounds which are found in living organisms are called biomolecules. These are the biomolecules. Organic compounds which are found in living organisms are called biomolecules. Before going to discuss about this, we have to learn something about living and non-living compounds which are present. There is a wide diversity in the living organisms. There is a wide diversity in living organisms. A question can arise in our minds whether all living organisms are made up with the same chemicals, that elements and compounds. There is a wide diversity in the living organisms. A question will arise in our minds. All the living organisms are made up with the same chemicals, that is elements and compounds. You have learned that in chemistry, chemical analysis process. Chemical analysis process. As we chemically analyze, as if we analyze chemically any living tissue of plants, animals or microbial paste, we will find a list of compounds. A list of compounds. So, which having the elements like carbon, hydrogen, oxygen, nitrogen and respective content of elements and element components in mass of the per unit. Mass of the per. Similarly, if we chemically analyze the piece of earth crust as example of inorganic, non-living item, the same compounds we will get from the earth crust also. Is there any difference between living components and non-living? Absolutely, there is no difference in both. Whatever the components are present, in non-living cells as well as in living cells. When a clear examination of non-living, if we clearly examination of non-living living things, so one thing, it consists of more hydrogen and carbon in non-living. We will find more hydrogen and carbon contents. Otherwise, so both in non-living and living matter, the same components are present. Same components are present. So then how do we chemically analyze the living components? How do we chemically analyze the living compounds? So if we ask continuously the question, we will find the answer. All the organic compounds which have, or living organisms which have made with the organic compounds. Organic compound. Similarly, if we analyze any living tissue of plants or liver, we will chemically analyze the components which are present in the living things. Just for that purpose, just take a piece of living tissue, either leaf or liver, and it is grinded with trichloroacetic acid with the help of mortar and pestle. So how do we chemically analyze the living organisms components? So just take a piece of any plant tissue, that is a leaf, or any animal tissue, a liver. Just the living tissue is grinded with the trichloroacetic acid with the help of mortar and pestle. So this grinded substance is filtered with the help of cheesecloth or cotton. We will get two components. One is filtrate. Actually, it is a acid soluble. The remaining is retent, which is non-acid soluble. So further in higher classes you will learn how to analyze chemically the components. So it is satisfied here that the extract subject to separation techniques till we get the one component, or otherwise we isolate single component. The separation techniques are applied to the extract till to get the isolated component. Then we will separate that. When we analyze chemically, we get an idea of molecular formula and molecular structure. When we apply the analyzing techniques to the extract, we get some idea of molecular formula and molecular structure of the compound. So thus, the organic compounds which are present in living organisms, are known as biomolecules. By analysis, whatever we get the organic compounds in filtrate, that the presence of organic compounds in living organisms is known as biomolecules. However, the living organisms also have inorganic components also. However, the living organisms have inorganic components also. So, for chemical analysis of when we done chemical analysis, we will get the inorganic compound. For that purpose, you take any living tissue as wet weight and allow to dry up. Thus, the water is evaporate and we get dry matter. The dry matter is completely burned. Thus, the organic compounds are oxidized and released into the air. Whatever the after burning, whatever the left, that is known ash. The ash contains inorganic elements like calcium, magnesium, inorganic compounds like that phosphorus, magnesium, sulfur. So thus, one can analyze the chemical components which are present in living organisms. So the living organisms which have the chemical components as sulfur, magnesium and phosphorus. So in view of chemist, the chemical components are functional groups: aldehydes, aromatic organs, aromatic components. In view of biologists, it is the amino acids, vitamins and lipids. This is the biomolecule introductory part. So what is biomolecule? This is the 10th chapter. So the organic compounds which are in the living organism, the organic compounds which are found in the living organism, that is a biomolecules. Before knowing the biomolecules, there is a wide diversity among the living organisms. The question will arise: all the living organisms are made up with the same chemicals, that is elements and compounds. You learned that the chemical analysis in chemistry. So similarly, any living tissue of plant, animal or microbial based or chemically analyzed, we will find some of the organic compounds. List of organic compounds of carbon, hydrogen, oxygen, etc. and its content of organic compounds per unit in mass per unit. So similarly, if the earth crust is chemically analyzed as example of non-living, the similar list we will found. So is there any difference? Means absolutely there is no difference. Whatever the components are present in the living organisms, the same are present in the non-living organism. But when we observe, the clear examination reveals that the living organisms having more hydrogen and carbon. So thus this is the chemical analysis to know the compounds. So tomorrow we shall continue this topic. This topic. So about the biomolecules. So biomolecules for 2 marks and 4 marks important. 2 marks and 4 marks important. So why have chosen these topics to explain to you for the first time means, so cell biology is something you know. You learned about cell biology in your lower classes. Because of that, I chosen the 9th chapter, cell structure, basic life of cell is the basic life of organisms, though that we completed in 9th chapter. Today we started that biomolecules. So in this, the most of the expected questions are 2 marks and 4 marks. 2 marks and 4 marks. So in further, we are going to discuss about that fatty acids, amino acids, lipids, and proteins, etc. So tomorrow we are going to discuss about that amino acids. What is amino acids? What are the functions of amino acids? How many types of amino acids are present in the living organism? Living, at the same time, the fats. What is fat? What is the soluble fats and insoluble fats? About that, and same time lipids also, and further the starch and secondary structure of the nucleic acid also we are going to discuss. Just go after completion of the topic, just you go through the topic and you will get an idea, and we will understand whatever the content is present in this, whatever I have explained you, so that way you will get an idea and easy to understand. So every day, if you go through the chapter at home, you will get definitely good result. Okay, here we are concluding the class. Thank you.
మీ స్కూల్స్ మరియు కాలేజ్ విద్యార్థుల కోసం ఆన్లైన్ డిజిటల్ క్లాसेस ద్వారా బోధించాలనుకుంటున్నారా?
क्या आप आपके कॉलेजेस, स्कूल्स और ट्यूशंस को डिजिटलाइज करना चाह रहे हैं?
మీ విద్యార్థుల కోసం సరైన ప్లాట్‌ఫామ్ వెతుకుతున్నారా?
क्या आप ऑनलाइन क्लासेस के लिए किसी बेहतर प्लेटफॉर्म की तलाश में हैं?
అయితే మా ఏఎన్ ఛానల్ ద్వారా ఉమ్మడి మహబూబ్ నగర్ జిల్లానే కాకుండా గద్వాల, వనపర్తి, నాగర్ కర్నూల్,